I'm Matt David, and I'm going to be talking with you about the pathways of descent in endogenous retroviruses. ERVs are the descendants of ancient exogenous retroviral infections, which made their home in our nuclear genome. So, what are ERVs fundamentally? Are they pathogens that we should keep repressed? A lot of research has focused on identifying their role in disease, especially diseases of autoimmunity, cancer, and neurodegeneration. However, a growing body of research is identifying fundamental roles that ERVs play in reproduction, pluripotency, and healthy normal physiology. One way that we can approach this is to follow the pathways of descent through an ERV that has begun to populate and be selected within its target host genome. The approach that I'm presenting here treats them as replicators which have crossed over from the kind of parasitic independent replicators whose fitness is not tied to the ultimate well-being of the host. These ERVs have crossed over, and now, because they reside within the host, their contribution to fitness means that they actually play a role in host fitness, and host fitness plays a role in their replication success. Following along with the graphical abstract, we can follow as an exogenous retrovirus makes its way into the genome. It now occupies space in that genome and begins duplication and transposition and starts occupying an increasing number of loci. What potential outcomes could these descendant ERV-derived elements have? One potential pathway would be extinction. The next major pathway could be coding acceptations, in which the protein coding DNA from the retrovirus is used to create peptides in the host. This could be initially pathogenic, or it could contribute to host fitness. Of course, if it does contribute to host fitness, this is a quick way for an ERV to achieve a selective advantage. We think this is the case in the case of some of the envelope proteins that have taken hold in the human genome, especially for classical cases like the contribution of multiple retroviral envelope proteins that contribute to placental morphogenesis in human reproduction. To highlight this method of following pathways of descent for our research into potential pathophysiology is to ask the question, what could go wrong with a fusogenic immunosuppressive peptide if it were expressed in the wrong environment? And we actually have an answer in the case of ERVW, the one that encodes syncytin in humans. Now, of course, there are multiple different syncytins in humans, but ERVW is the most well-studied, and it is now known to contribute to the pathogenesis of multiple sclerosis. We can apply the same logic to the other viral peptides as well, both to ask what their contribution to healthy physiology would be and what their potential pathologic implications would be as well. The next major branch would be adaptations for the non-coding contributions of ERVs, like the long terminal repeats that flank ERVs, which are known to contain transcription factor binding sites and alternative promoters. We now know that these play a huge role in gene regulatory networks, in other alternative promoters that allow the genome to become more adaptively robust, and we know that they contribute to a significant number of non-coding RNAs. We can also ask about what the contributions would be from a growing number of ERVs scattered throughout the genome to our chromatin architecture and DNA packaging. The close correlation between the presence of ERVs and the presence of silencing mechanisms now shows us the role that ERVs play in chromatin architecture and DNA packaging. So those are our pathways of descent. Pretty much all ERVs will fall under one of these coding or non-coding pathways. The pathophysiology and the potential for acceptation, adaptation towards the fitness improvement of the host, are available within these pathways of descent. In the center column of the poster, you'll see 3 of the steps of this endogenization process described in a little bit more detail. Endogenization, it appears, occurs primarily because there is a selective benefit that happens in a sort of leap under the pressure of exogenous retroviruses. This appears to happen because envelope protein allows superinfection prevention by receptor interference, when the endogenization is actually happening. Essentially, one XRV, one exogenous retrovirus, is able to outcompete its cousin specifically by getting inside the host cell and then shutting off the host cell as an access point for others. It then wins the replication game by already being inside the host, and then it's able to propagate, oftentimes outliving the exogenous retroviral cousin. As an exapted protein makes its way into the genome, and over time, when the rest of the genome adapts around that, the role that it serves can cause the host to undergo induced dependence. A classic example of this is, again, ERVW. By generating syncytin, it increases the placental invasiveness, increases the nutrient exchange, and protects the fetus from immune attack by the mother. And at that stage, the host is now dependent on that endogenous retroviral envelope protein to perform its exapted role. And if we were to take it away, the fetus would not survive and reproduction would not be possible. Thus, induced dependence means that once these retroviruses are an integrated part of healthy, normal physiology or reproduction, it's no longer possible to simply remove them. This, of course, becomes highly problematic when we identify that that same feature becomes a trade-off adaptation. That is, that adapting to include it in normal physiology means that it provides another alternative disease pathway. Understanding ERV pathways of descent helps us understand that these trade-off adaptations don't automatically make the ERV contribution a pathogenic one, but that it may serve a pathogenic role even if it is serving a healthy, normal physiological role in a different context. And of course, that makes diagnostics and therapeutics a little bit more challenging than it would be if the virus were simply an exogenous pathogen. Understanding these ERV pathways of descent means that we have to take into account the fact that especially the ERVs that have been with us for a long time and do play exaptive roles in our physiology can't be treated simply as an exogenous pathogen that we can simply attack or remove. Instead, we have to recognize that the pathogenicity comes from expressing it in the wrong tissue at the wrong time or in the wrong way. Understanding the way that these ERVs came to be present and distributed in the genome can offer us insights into why we see such a strong contribution to reproduction, to pluripotency, and to the particular categories of disease that have received the most attention so far. The takeaway from this model is that ERV research needs to make strides in establishing baseline expression levels across the sexes, across different tissues, across different ages, and across different patient populations to determine what the normal physiological role of ERVs is. And then from there, we need to build out profiles on their interactions with diseases and, of course, with pharmaceutical agents. Studies should include the presence of ERVs, the copy number, variants, and of course, they should include activity, methylation status, and any other changes to the profile of ERV activity and expression and repression. We should understand that since some physiological roles are played by ERVs, sometimes pathogenesis will occur because ERVs are underexpressed, sometimes because they're overexpressed, sometimes because alterations in the methylome have happened because of interactions with the environment, with other disease processes, or even with exogenous viruses who are able to activate them through interactions with their transcriptional factors. Therapeutically, we have to understand that because ERV expression plays different roles at different tissues and stages, that the interventions that may be available to one age group or one population group may not be effective or may even be dangerous for another age group or another population. As we are in an age of advanced biotechnology, including CRISPR and rapid vaccine development, it's important that we establish these baselines before making the kinds of leaps and bounds in therapeutics that would allow us to engage with ERVs and their pathologic role. And that is endogenous retroviruses, pathways of descent. I hope you enjoyed it. Remember, we are more ERV than we are mammalian genes, so this is an absolutely vital piece of future research. I'm really excited to see where we'll be going.